Okay, let's move on to photosystem 2. That is the first of these photosystems that oxidizes water to oxygen. It is a real monster. It is a membrane protein. It has a total of 19 subunits, 36 molecules of chlorophyll A, 7 carotenoids, and beta-carotenes, and that's its dimensions. This is just a cartoon of it here. But you can see the basic plan, and we'll have more detail on this a little bit later. There is this special pair of chlorophylls right in there called the special pair of chlorophyll A molecules, and their interaction, their electronic interaction between them allows them to have a lower excited state energy and to capture the photon of light from all of the other chlorophylls in the molecule. Once they've captured that energy of the light, they use that to kick an electron out. It goes through another chlorophyll, a pheophyton, which is a chlorophyll without a magnesium, a quinone in the QA site. It's transferred to another quinone in the QB site, and then that quinone enters the quinone pool out in the chloroplast thylakoid membrane. And it's actually a slightly different form of quinone used in plants called plasmaquinone, and it's a little bit different structure from the quinone we found in the mitochondria, but the same basic structure. Meanwhile, we generate an electron hole that's highly oxidizing on this system, and that pulls electrons out of what's called the oxygen evolving complex, which is a cluster of 4 manganese metals down here, and that is able to oxidize 2 waters to oxygen. That's a blow-up of this structure down here. That cluster contains 4 manganese and a calcium, and that electron transfer chain pulls the electrons out of there. Now it takes the removal of 4 electrons from 2 waters to produce 1 oxygen, and this goes through a little cycle. Nothing happens until you've removed all 4 of the electrons from that manganese cluster. So you don't produce oxygen until after the 4 electrons have been removed. The removal of each electron requires 1 photon. So if you start with the reduced state S0, the first photon removes 1 electron, the second photon removes the second electron, then the third, and then the fourth, and then and only then is oxygen produced. So it takes 4 photons to remove 4 electrons from this center. The electrons are sent off here to the quinone pool. And finally, after that 4 photons, we produce oxygen. And we can see this in classic experiments. You do an experiment where you flash it with 1 flash of light, 1 photon, nothing happens, no oxygen. Second, nothing. Third, nothing. The fourth one allows the oxygen to be produced. So we need to go through that 4 photon step to remove the 4 electrons and produce that oxygen molecule. Here it is. This is the monster. This is photosystem 2, and this, again, was a remarkable achievement. It's a huge membrane protein. This is the whole structure. It's a little difficult to see here. This is actually a dimer. 1 complete molecule here, another complete molecule there. The one thing you can see in this diagram here, lots of alpha helices crossing the membrane. That is the major motif of membrane proteins. We have all of these transmembrane alpha helices that are particularly well suited for existence in a membrane environment. This is the inside of the thylakoid, the lumen, that's the outside, the stroma. All of these are chlorophylls that act as antennae. They capture that photon of light, they transfer it randomly amongst themselves until it finally ends up in the special pair, which is right in there. It's hard to see here. We'll see that a little bit later. And then the electron is removed from the oxygen evolving complex, OEC. That's where the oxygen is produced. The electron goes up here and reduces the QB and the quinone pool. Okay, let's bore in on this a little bit more. This is a view from the top of the the same molecule, but we're only looking at one of the monomers of the dimer here. And this is the central part of that molecule. There is that special pair right there, the two chlorophyll molecules that are they're coplanar but offset, about like that. And that's where they're located. You can see the quinone A, the quinone B, the oxygen evolving complex. Where is that? Don't see that at the moment. It's in there somewhere. Maybe it's not in there. Then out here, there's other subunits with antenna chlorophyll molecules. If any one of these captures the light, it is transferred from one to the other. Finally, when it gets there, it is trapped. That has lower energy. It traps it and initiates the electron transfer cycle. This is a more useful picture that just focuses in on the most important of these. Here is the special pair right there. That's what captures the electron. Here's another chlorophyll where it captures the photon. So the photon excites this special pair, kicks an electron out. It goes through chlorophyll A, the phaeton up there, which is a chlorophyll without the magnesium, then to a quinone called QA, then the QB, and then the QB, which is now reduced to QH2, leaves out into the membrane. It leaves an electron hole in the special pair that pulls an electron out of the special tyrosine, creating a highly oxidizing tyrosine radical. That in turn pulls an electron out of the oxygen evolving complex. As we go through 4 photons, 4 electrons are sequentially going up that way, pulling 4 electrons sequentially out of the oxygen evolving complex. Finally, then, we have enough oxidizing power to oxidize 2 molecules of water to 1 molecule of oxygen. So 4 photons are required to successively excite that special pair, kick electrons one at a time up there, and pull the electrons one at a time out of the oxygen evolving complex. Again, 1 photon, 1 electron. That's always the stoichiometry that we have here. Okay, here is that special pair that we focus right in on. And I can't emphasize the importance of that thing too much. That geometry allows the electronic structure to interact between those 2 chlorophyll molecules, monomers, in the special pair, to have a lower excitation state energy and to capture that photon from all of the other antenna chlorophylls. The total number of antenna chlorophylls is over 1,000 from other antenna pigments that are in the system. They all wander around very rapidly on a picosecond time scale until they finally get to that special pair, and then they are trapped by that lower energy. So that special geometrical orientation between the chlorophylls and the special pair is ultimately responsible for all energy production and all life on this planet. So that geometry is a very special type of geometry that you see right there. And people have analyzed the physics of that in great detail. By the way, this is a beautiful thing to study as a physicist as well as a chemist or a biologist, because it has some of the fastest reactions that are known. So high-speed physics is a very useful method to use to study the system. Okay, here's just another blow-up of a picture that we've seen before, reminding you what happens again. The photon of light comes in, excites the special pair. The electron goes through the chlorophyll up to the phaeton. That occurs within a few picoseconds, one of the fastest reactions in physics. It then goes up to the QA in microseconds, over to the QB, and out to the Q pool. So every electron goes up that way. Meanwhile, we create this electron hole here. It pulls an electron from the tyrosine. That pulls it from the oxygen evolving complex. Once we've pulled 4 electrons out of there with 4 photons of light, we can oxidize the 2 waters to 1 oxygen. So that's one of the things we want to understand here. Now, this is kind of a complicated picture, but let me explain what this means. This is an energy diagram. That's just the energy vertically here. That's the energy of the special pair in its ground state. That's the energy of the special pair of chlorophylls in its excited state. These give the lifetimes for the movements of the electron to the different systems that have to occur, as I've shown in the previous slide. To move the electron from the special pair to the phaeophyton takes just a few picoseconds. To have the electron go on to QA takes about 200 picoseconds. Now, look at this reaction here. The natural thing that could happen here is the electron in the phaeophyton could come back to the positive charge in the P680, bring you down to the bottom at the beginning. That would be a futile cycle. Nothing good would happen from that, and that would limit the efficiency of photosynthesis. That is the major problem that limits the efficiency of solar energy cells, for example. It's the same process that limits the efficiency of the ruthenium technique that we use in our laboratory. So that's the main holdup to high efficiency. What the natural photosynthesis system has done here is that it's set up the geometry of that site in the active site so that reaction takes place in 200 picoseconds, but the back reaction that would limit the efficiency takes place 1,000-fold more slowly. So really the secret of photosynthesis is making that forward reaction much faster than that back reaction, which would ruin the whole thing. If we understood how that would work and could make a solar energy cell with that type of efficiency, we would solve our energy problem right there. The plants do this with 99% plus efficiency. It's a totally amazing process. The next thing that happens is we pull electron out of that tyrosine in the oxygen evolving complex. That's 50 nanoseconds. It depends on what part of the cycle we're in. We then transfer the electron to QB in 100 microseconds. We then complete the oxidation of the manganese, or begin the oxidation of the manganese in the microsecond time scale. All of these could potentially have back reactions which could kill the whole process. But all these back reactions are slow compared to the forward reaction. So the secret of photosynthesis that we still don't really understand completely is how these forward reactions are so fast compared to these backward reactions. And an understanding of this would be really helpful in creating better solar energy devices. Okay, now here is another picture of that cycle that is needed to produce oxygen. And we start out with the most reduced state. One photon pulls out one electron, making S1, which is more oxidized. Second photon, S2, more oxidized still. Third photon, S3. Fourth photon, S4. And then we have the complete oxidation of two waters to one oxygen. Remember, that takes the removal of 4 electrons. They are removed one at a time from the manganese cluster, and then, and only then, do we produce the oxygen molecule. Now, just in the last year, we have an improved X-ray crystallography spectrum here, a structure here, where we now have a pretty good picture of what this manganese cluster looks like. And it has 4 manganese, which are colored purple, as you can see here. It has 1 calcium that is colored yellow or green, I guess. And then the other positions are occupied by oxygens. This forms a cubical type of molecule. See, that's a cube with calcium at 1 apex of the cube. The manganese, 3 manganese are at the other apex. And then there's here an extra manganese that's sitting out there. We now believe that the region of the oxygen evolution is between the calcium and this extra manganese, right in there. And other parts of the protein help to maintain this particular structure and assist in this very difficult reaction of the oxidation of water to oxygen. here is a current model of what it looks like. I should emphasize that this is really a model or a hypothesis. There's quite a bit of evidence that favors this, but it's not yet complete. And here's the cluster, and the free manganese in the cube, the extra calcium, and here's the extra manganese off to the side. That extra manganese binds one water, the calcium binds another water. And all the manganese are fairly reduced here, and we pull out one electron with the first photon, and notice we oxidize that manganese from +2 to +3. That's the first oxidation. We pull out another electron with the second photon, we oxidize that manganese from 3 to 4. Pull out the third electron, and we oxidize that manganese from 3 to 4. And as we're going along here, we're removing protons as well. Notice we removed one proton there, another one here, we removed the proton there. Then we pull out another electron, bring that manganese to a 5 oxidation state, which is pretty unusual. And then we have the attack of the OH on the calcium on the oxygen on the manganese, and we form this critical oxygen-oxygen bond. That is the critical step in this reaction. And then finally, that is released, another proton comes in, that's released as oxygen, as you can see there, another proton is released. That completes the cycle. So we start out with the two waters on the cluster. We successively remove the electrons and oxidize those manganese atoms until we get to this state. And then we allow the oxygen, the water attached to calcium, to react with that manganese and form that oxygen, which is then released. So 4 photons successively remove 4 electrons, and then finally that leads to the oxidation of the 2 waters to the single oxygen. Now, as I mentioned, there's a lot of, this is just a hypothesis, there's a lot more that needs to be done. One of the most exciting things that I saw in Germany at the conference that I went to in September was that a group has developed a new technique where they can actually take time-resolved X-ray crystal structures as they are going through this very rapid mechanism on a picosecond time scale. It's an amazing new technique. And they're hoping to use that to get improved structural information to provide additional evidence for this mechanism or an alternative mechanism if it's found that this one is incorrect in certain details. So the ability to get structural information at nanosecond time scales is really amazing, and that was one of the most amazing things at the conference. Okay. Coming back now to our basic plan here, what we've just talked about is this photosystem 2. We have oxidized the water to oxygen. We have reduced the quinone to QH2. We now are going to go through the Q cycle using B6F, which is completely analogous to the BC1. The electron is then carried by plasticyanin over to the photosystem 1. That then carries the electron to ferredoxin reductase, and that reduces NADP to NADPH. We also have a good crystal structure of this photosystem 1. So let me just look at that. Here it is. Again, a large membrane protein, and we have here lots of transmembrane alpha helices. The special pair is right down about there. We have all of these iron-sulfur clusters which act as electron transfer centers. Now let's look at the innards of this thing, and that's in this next slide. Here is the special pair down here. Nearly that same geometry we found in photosystem 2, except this is excited at 700 nanometers, slightly lower energy, longer wavelength. Once the photon gets to that position, excites that, the electron goes to this chlorophyll molecule up to a phi of 10. with quinone there, to an iron sulfur cluster there called FX, another iron sulfur called Fa, another iron sulfur called Fb, and then out to paradoxin. So that's what this thing looks like. Just one more slide, and here's the plan again. And I'm now going to look at the structure of this B6F. This is what it looks like. Unfortunately, they printed this upside down compared to what we had before. But it contains the cytochrome B on one side, another cytochrome B on the other side. It contains a plastic quinone site on each side of the membrane. There's a cytochrome F out here, the plastic cyanide. This has nearly the same type of function and structure as the cytochrome BC1. It uses electrons through a Q cycle to pump protons across the membrane with the same stoichiometry. 2 protons are pumped per electron, and so that would pump a total of 8 protons for the 4 electrons that go through here during the complete scheme. Okay, so I think that's where I wanted to get to today, and so we'll continue.